I believe it's after 11 o'clock if you guys are ready to get started. Are you all able to hear me online okay? Good. Cool. The Center for Integrative Environmental Health Sciences and the Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar today. Dr. Alexandra Nell is a T32 postdoctoral fellow in the lab of Dr. Chris States. Her work focuses on identifying molecular mechanisms that influence DNA double-strand break accumulation in cells exposed to arsenic. Dr. Nell received her BS and MS in biology from Morehead State University and her Ph.D. in microbiology at the University of Kentucky. Her PhD work focused on both zinc finger and homobox gene duplication throughout chordate evolution and post-transcriptional regulation of duplicate genes CYP2A4 and CYP2A5 in the mouse liver by two nuclear-enriched, alternatively spliced, long non-coding RNAs that she identified and characterized. Since joining the States Laboratory in January of 2021, she's published 3 manuscripts, including a chapter review in Genomic and Epigenomic Biomarkers of Toxicology and Disease, Clinical and Therapeutic Actions, and co-author reviewed article published in Seminars in Cancer Biology. Her work that she's sharing with us today has recently been published in the Journal of Toxicology and Applied Pharmacology and served as the basis for her recently submitted F32 application entitled DNA Double-Strand Break Repair Pathway Choice in Human Keratinocytes Following Arsenic Exposure. Among her awards, this year she won the Society of Toxicology Dermal Toxicology Specialty Section Stratocorp Postdoctoral Award, First Place Postdoctoral Award for the Society of Toxicology Graduate Student Leadership Council 3-minute thesis competition, and won first place at Research Louisville in 2021 for her poster presentation. Dr. Nell is highly qualified academically and a pleasure to work with. With that, I'll welcome Dr. Nell to present today's seminar entitled Chronic Arsenic Exposure Reduces DNA Damage Response Activation in Human Keratinocytes.
All right, thank you so much, Kennedy, for the nice introduction, and welcome everybody to the CIEHS Pharm Talks seminar series. This is actually my first seminar since 2019. So it's a pleasure to be here, and I'd like to thank everybody for the opportunity. So a quick outline of what I'll be discussing today. We're going to go over a bit of a background on environmental arsenic exposure, some human diseases associated with chronic arsenic exposure, arsenic and skin, proposed mechanisms of arsenic-induced carcinogenesis, and I'll touch on chronic arsenic exposure and the DNA damage response as a review, and I'll get into my hypothesis, my models, my methods, and the results from my study to end the lecture. Can you see me okay? Okay. All right, good. So arsenic is known as the king of poisons. Typically, when you talk about arsenic, people associate this with a long history of homicide. And so it's tasteless, odorless, and colorless when dissolved in liquid. And a famous example of somebody that was poisoned by arsenic, or potentially poisoned by arsenic, was Napoleon Bonaparte. And so samples from his hair actually show high concentrations of arsenic. And a more recent example for poisoning was actually in Bangladesh, where greater than 30 million people have been poisoned by arsenic in their water supply after they switched from drinking surface water to digging tube wells. And so these tube wells have high concentrations of arsenic in them, and this has led to a mass poisoning. And in general, I always like to talk about the history of arsenic because I find it really fascinating. And this actually just came out in April in National Geographic, where they've actually found that old books that were wrapped as an alternative to leather-bound books actually contained high levels of arsenic in their bindings. And the reason for this is because they're tinted with Paris green. And so Paris green historically was used, from its name, you can tell it was used predominantly in Paris. The Paris was covered in green, this color of green. And so it was used in wallpapers, dress pigmentations, and so people would get exposed to arsenic because it was actually being used to pigment these different textiles. And in addition, arsenic has a long history of being used for beauty supplementation to try and clear up complexities of people. And it's also had a medicinal purpose as well, but has later been found to actually cause chronic arsenic poisoning from people that were exposed in these ways. And this is a little bit more recent, but arsenic used to be used in pesticides as well. And so a lot of the different cotton fields or tobacco fields where they used pesticides that contained arsenic have since now been utilized as rice fields. And so this kind of gets into more of how we're exposed in today's world. And so in industry, people are, especially in copper smelting, people can get exposed to arsenic through inhalation, and so there's a high association with lung cancer. You can, and like I just mentioned, different products, different crops where we've sort of reutilized the fields for different purposes in different crops. You can be exposed to high levels of arsenic in different foods. And by far, the most common route of chronic arsenic exposure is from drinking water supplies. And so this is a picture of someone in Bangladesh pumping their water from one of the tube wells. Now, there has been some remediation efforts to try and mark wells that are highly contaminated with arsenic, and so these have been marked in red now. However, people still will utilize these wells because it's the only water for miles. So they're well aware that they're being poisoned, but there's nothing that's really can be done about it. And this is something I found out more recently. If you look up arsenic in playgrounds, they actually found that a lot of the lumber that was utilized for playground contains high amounts of arsenic in the treated lumber. And so this is a big case of it was definitely in Florida, where they had to, cross off all these playgrounds and rebuild because they found high arsenic concentrations and children were being exposed to very high levels. So as I mentioned, chronic arsenic exposure, it's a worldwide and it's a US problem. You mostly get exposed from drinking water supplies that are contaminated with arsenic. And so in general, it affects about 225 million people across 108 countries. From what I've seen in the literature, these numbers just keep growing as more data is being collected. In the US, about 3 million people are affected. So not as many, but it's usually from contaminated well water sources. And in the early 2000s, the maximum contamination level for arsenic in drinking water was set to 10 parts per billion, so this is about 10 micrograms per liter. And this was set by the EPA and the World Health Organization. Previous to that, it was 50 micrograms per liter. And it is estimated, so this is an estimate from 2007, that even in the regulated water systems, so this is outside of well water systems, about 4,000 different well water systems serving roughly 3 million people are likely to exceed this 10 parts per billion water standard in the US. So it's still a big problem even within our country. In terms of the region in Kentucky, so this map is showing the different geological formations, and then these dots here in red are showing well water with high arsenic concentrations. And typically these high arsenic concentrations are associated with coal mining activity. And so a big problem in Kentucky is that these coal mining areas generate significant levels of coal ash. And so this ash, it wasn't regulated before, and so they were pouring it into these unlined landfills, and they later were found to be seeping into groundwater sources. And so these are some pictures showing the water runoff and the pollution around these areas. And so this is typically this orange color can be seen in areas that have high amounts of this coal ash pollution. So this was an article that came out in 2017. If you're interested in looking at more information about it, you're more than welcome. I found it very interesting. And so in terms of diseases associated with chronic arsenic exposure, there are a myriad of different diseases. It doesn't only cause cancer, it can cause cardiovascular issues, gastrointestinal disturbances, it can cause diabetes, neuropathies, so in the nervous system. And the in general, arsenic is classified by the IARC as a group 1 human carcinogen. And so this is because it's established to cause cancer in multiple different organs. And in general, it's been #1 on the ASTDR national priority list of hazardous substances for over 20 years. And skin lesions are often the first signs of chronic arsenic poisoning. So up here at the top, we have skin. It can cause skin lesions and skin cancer. Over here, this is just showing a typical hyperkeratosis condition that's caused by chronic arsenic exposure, usually presents within 6 months to 10 years of being chronically exposed to arsenic in drinking water supplies. In terms of non-malignant skin manifestations of chronic arsenic exposure, they include hyperpigmentation, raindrop pigmentation, palmar and plantar hyperkeratosis. So these are in regions that are usually non-UV exposed. And so in general, these are hallmark signs of chronic arsenic exposure. In terms of malignant skin lesions, the hyperkeratosis can develop into Bowen's disease, which can eventually develop into squamous cell carcinoma. And also, the hyperkeratosis can directly evolve into basal cell carcinoma. And so skin is a major target organ of arsenic, so that's why we're interested in it in the States lab. And in general, this question always comes up about the comparisons between arsenic-induced skin cancer and sunlight-induced skin cancer. So in general, in arsenic-induced skin cancer, Bowen's lesions are seen very often. In sunlight-induced skin cancer, they're very rare. Squamous cell carcinoma can be seen or induced by both. Basal cell carcinoma can be induced by both, and malignant melanoma is often characteristic of sunlight-induced skin cancer. The one thing I will say is that in arsenic-induced skin cancer, you can have presentation of all of these different types at the same time. So that's something that's very characteristic of arsenic exposure. And so in general, a big question in the lab is what molecular mechanisms contribute to skin carcinogenesis? And so we know that in sunlight-induced cancer, there is unrepaired UV photoproducts, and so this typically occurs in UV-exposed regions. And so TP53 is a major target, and in exposed individuals, you'll have UV signature mutations, and so these are typically C to T mutations, and they're especially prevalent in TP53. They appear in all sunlight-induced non-malignant lesions, pre-malignant lesions, and squamous cell carcinoma. And in general, TP53 mutations are very rare in arsenic-induced skin cancer. And there's no UV signature mutations that are present in arsenic-induced skin cancer. And so just based on this information and based on the presentation of individuals that are exposed to arsenic, the molecular etiology is very different between these two different conditions. And so we're really interested in figuring out what are the molecular mechanisms that contribute to arsenic-induced skin carcinogenesis. And so before I get into our studies, I just want to mention that there are a lot of different proposed mechanisms for arsenic-induced carcinogenesis. This is not an exhaustive list. They include DNA repair inhibition, co-mutagenicity, epigenetics, including miRNA expression, which is another avenue of research in the lab, oxidative stress, cell proliferation and stem cell expansion, differential alternative mRNA splicing, and also aneuploidy and chromosomal instability, including both numerical and structural chromosomal abnormalities. So in general, my research focuses on these two areas, DNA repair inhibition and chromosomal instability. And so I'll next get into talking about sort of some data that really set up my experiments and study. So before I got into the lab was interested in trying to figure out which molecular mechanisms contribute to skin carcinogenesis. And since all of these different really mechanisms have been proposed, we wanted to try and figure out the different gene expression changes that might contribute to this. So we performed RNA sequencing. And the way that we set the experiment up is that we had HACAT cells, which is a well-studied model for arsenic-induced carcinogenesis. And we cultured them in independent quadruplicate cultures. So we had 4 independent dishes that were either unexposed to arsenic or exposed to arsenic. And we collected at different time points. So this is 7 weeks, 19 weeks, and 28 weeks. And we performed RNA sequencing on these different time points. And in addition to that, we also looked at exposure initiation, so the transformation process of these cells. And so we've published a paper on this, and so we can see from this paper and our studies that we conducted that there are early changes associated in transformation. Transformation initiation doesn't really begin until 19 weeks chronic exposure. And cells are fully transformed, and we did this through EMT marker characterization. So they're fully transformed by the time we get to 28 weeks. And so between each of these time points for unexposed versus arsenic exposed, we performed differential gene expression analysis to try and figure out what are the molecular changes that are occurring in cells at these different time points after being exposed to arsenic. And so for my studies, I'm really interested at this 7-week time point because these are early changes in during the transformation process. And so I think that these are important in trying to figure out what are the early changes that lead cells down the road towards cancer development and transformation. This is a snapshot of some ingenuity pathway analysis that other lab members performed after this RNA sequencing experiment. And so this is a huge pathway. I know nobody can probably read it, but if we look at this blow up over here, what we found from this pathway analysis is that the ATM signaling pathway was dysregulated. And one thing that I really want to point out about this pathway is it's a very apical pathway, and there's also interconnectedness between all the different signaling pathways and DNA repair pathways present in the cell. And so that can kind of be seen by all these lines denoting all these connections. But the big thing that we found from this is that ATM signaling was dysregulated. And before I get into talking more about my research, I do want to briefly discuss the DNA damage response. And so there are 3 apical kinases that really signal when DNA damage occurs in the cells. And these are DNA double-strand breaks. And so you can have endogenous or exogenous DNA double-strand breaks occurring, which can activate DNA PKCS or ATM, depending on the cell cycle. And downstream of DNA PKCS and ATM, as well as ATR, is P53. So everybody knows this as the guardian of the genome. So when these apical kinases are activated, they signal to P53, which halts the cell cycle, or if there's enough damage, can induce apoptosis. Downstream of ATM, there are also check kinases. So this includes check2, which can signal to CDC25A, which if the cell is in S phase, it will halt the cell in S phase in order for DNA damage to be repaired. Downstream of ATR is check1, which can signal to CDC25C. So if the cells are in G2, then it halts them in that point until the DNA damage is repaired. And so all of these different apical kinases that make up the DNA damage response really cooperate throughout the cell cycle in order to prevent DNA damage from occurring in cells. And so in general, the important points to keep in mind are that normal cells, they can either fix their damaged DNA and continue through the cell cycle, or they die if the DNA is not repaired. And so if you have cells that continue throughout the cell cycle with DNA damage, then you can pass this damaged DNA to daughter cells. And defective genomic integrity is a hallmark of carcinogenesis. So cells that have this DNA damage can then pass this on to daughter cells, which then can lead cells down the pathway of becoming cancerous. And in general, these different apical kinases are initiated by different factors. So DNA PKCS is actually initiated by KU complex. And so these initiation complexes are important for recognizing the DNA double-strand breaks or the stalled replication forks and then signaling downstream to these DDR apical kinases. And so for ATM, MRN is the initiator, and for ATR, ATRIP, and RPA signals to ATR. And so this, again, this is sort of going over what I just covered, and is that downstream, if you have these kinases activated, then you can get DNA repair, cell cycle control, which cells can be halted in order to repair the damaged DNA. Cells can actually undergo senescence in addition to alternatively apoptosis, depending on how bad the damage is. What is also known about arsenic is that it's a clastogen. And so in general, a clastogen means that double-stranded breaks are occurring in cells, so an agent that induces double-stranded breaks or causes an accumulation of double-strand breaks. And so this is a picture from isolated lymphocytes from a chronically exposed individual in Mexico. And so the blue arrows are pointing to gaps in a chromosome. Hopefully you all can see that. The yellow is pointing to an acentric fragment from another chromosome, and the black is pointing to an isochromatic fragment. And so all of these are indicative of double-strand breaks occurring in cells. And this has been established both in vitro and in human exposed populations. And so something else that's known is that arsenic is weakly mutagenic, so it does not form adducts, it does not interact with, directly interact with DNA. And so it's quite unusual in regards if we're comparing it to other chemical carcinogens. And the last point is that it also enhances the mutagenicity of other carcinogens as well. And so this is a quote from the Arsenic in Drinking Water textbook from 1999. And so it's in general accepted that the most likely explanation for the mode of action for arsenic carcinogenicity is that it induces chromosomal abnormalities without interacting directly with DNA. And so this is, from 1999, so this is a long-standing issue that still hasn't been solved for how arsenic is actually causing accumulation of double-stranded breaks. So my major question that I want to answer is, how is arsenic exposure causing the accumulation of double-strand breaks? And so in general, I think people forget that double-strand breaks can occur endogenously in cells every day. And so these are estimated to occur between 10 and 50 every day, and that's dependent on cell cycle and tissue type. And an agent can either induce DNA damage directly, so this is an exogenous agent, can either induce DNA damage directly, or it can prevent repair of DNA damage in cells. And so both I guess, could lead to the accumulation of double-stranded breaks. And so the question becomes, is arsenic inducing double-stranded breaks, which is unlikely because we know that it doesn't directly interact with DNA, or is it inhibiting double-strand break signaling and repair? And so the latter is what I've chosen to base my studies on. And so now I'll get into sort of setting up what I've been working on for the past year. So my hypothesis is that chronic arsenic exposure results in reduced DNA damage signaling, promoting double-strand break accumulation, which contributes to arsenic-induced chromosomal instability. And in terms of the NIEHS translational framework, I want to put our studies into perspective here. So the ultimate goal within this framework is to change outcomes for different populations that might be exposed to arsenic, change the environmental exposures, change clinical outcomes, and change economic outcomes. And so when people are chronically exposed to arsenic, there's not only health issues, but there's socioeconomic issues because they're sick. And there's a bunch of other things that can occur. And so it's important to try and study this in terms of how it might affect a broader audience, I guess. Sometimes I feel like as a trainee, I don't always think about that when I'm doing experiments in the lab. And so in terms of policy, the EPA and IARC, they designated arsenic as a group 1 carcinogen. And so what we've really focused on is looking at group and population human characteristics and then trying to translate that in the lab by looking at human cells in vitro. And so in general, the group and populations for humans, I've already discussed this. The observation is that chronic arsenic exposure induces skin carcinogenesis because skin is a primary target organ. In terms of the identification, there have been many studies that have shown that in vitro with human cells, chromosomal instability is identified as the major driver as arsenic-induced carcinogenesis, which was also mentioned from the 1999 textbook. And so in order to better understand how this chromosomal instability might be occurring, we're also looking in vitro at human cells. And our question really is, how does chronic arsenic exposure cause structural chromosomal instability? And the significance and innovation for the studies that we're doing in the lab, they utilize toxicologically relevant arsenic concentrations. And so we've modeled our experiments based on mean blood arsenic concentrations that are found from human exposed populations. We have chronic exposure conditions, so this isn't always the case. A lot of studies in the field have done acute exposure conditions, so only hours to days, and they've utilized very high arsenic concentrations. And then, of course, in our lab, we're interested in human skin cell lines because that skin is the major target organ, and we utilize multiple different ones because it provides robustness to our studies. So the different skin cell models that we use include HACAT and KIR-CT cells. HEC-N have also been used within the field. So I want to just dive into a little bit why we use these different models. And so the origin for HACAT cells, they actually came from a margin of malignant melanoma. So they're not malignant melanoma, and they've actually been spontaneously immortalized. All of the cell lines that we use are from males. I just want to point that out before I forget that. And in KIR-CTs, they actually are derived from neonatal foreskin. HEC-Ns are as well. The difference between these is that KIR-CTs have been immortalized with hTERT and CDK4. In terms of how well studied these are, HACAT cells and HEC-N are the most well studied. KIR-CT, in terms of chronic arsenic exposure, it's a model that we're developing currently in the lab. Replicative senescence, so this is probably the biggest determinant of why we're using the cell lines we're using for chronic studies. So both HACAT cells and KIR-CT cells, they don't have replicative senescence, whereas HEC-N actually senesces after 15 or 16 passages, and so they're not they're hard to use for chronic exposure studies like we're conducting in the lab. In terms of the karyotype, the KCAT cells are pseudotetraploids. They, on average, have about 77 chromosomes. KIR-CTs are near diploid. They contain between 45 and 48 chromosomes, which makes them ideal for karyotyping. And HEC-N are diploid, but again, we can't really use these for chronic exposure. So in the lab, we typically utilize HACAT and KIR-CT cells. And so this is how we've set up our different conditions. So we have HACAT, either unexposed to arsenic, sodium arsenite in this case, or 100 nanomolars of sodium arsenite. And we've exposed them up to 28 weeks. So this is from that study that I described earlier until we can see EMT occurring. And so this is the setup. We continuously passage them for unexposed or for exposed, keep them independent from one another, and we keep them up to 28 weeks this way. And at the 7-week time point, this is that early change time point that I'm interested in. For my studies, I performed immunoblotting and conducted neutral COMET assays to look at double-strand break accumulation. And I'm developing a new model for chronic arsenic exposure using KIR-CT cells. We've essentially done the same exact setup. The only difference is that we've isolated at an 8-week time point compared to a 7-week time point, but I've conducted the same types of experiments for these cells as well. And in addition, since this model is in development, I've looked at double-strand break accumulation after 32 weeks of chronic arsenic exposure. And in terms of the COMET assays, I like to review this because some people aren't familiar with it. And so COMET assays in general, this is also known as a single-cell electrophoresis assay. They can be used to assess either single-stranded or double-stranded breaks. In my particular studies, I'm using neutral COMET assays. And so this is a general schematic of how you set up a COMET assay. So you can have samples from whole blood or other tissues or cells. You can have cultured cells or even frozen cells if you've worked up the conditions correctly. And you isolate these cells into single cell suspension, put these cells into low melting agarose, put them onto slides, lyse them in a high pH buffer for 1 hour, then you electrophorese them, and then you precipitate the DNA onto the slides, and then you can stain them with various things, lithium bromide. In my studies, I've used cyber gold. And then you can image these slides afterward and analyze to see all the double-stranded breaks. So this is just an example, and I hope it's pretty obvious what we're looking at here. So unexposed cells, so this is showing, I think the biggest thing to keep in mind are these red circles. And so we can see that in the unexposed cells, these red circles are kind of tight around this white dot. And in our arsenic exposed cells, you can see that these red circles sort of expand. And so this is indicative of DNA damage occurring, and this would be a positive for DNA damage. We also run positive controls with our experiments. So neocarcinostatin, which is a double-strand break-inducing agent, or hydrogen peroxide is another good one. And there's a number of different positive controls that you can use depending on what you want the readout to be. And so for my experiments, I'm going to be quantifying this olive tail moment. And so this is showing a comet. And so this is considered the head of the comet, whereas the tail is that broken DNA that's migrated out after electrophoresis. And so the olive tail moment is this tail length here, and then the percent DNA, tail DNA throughout the whole comet. So you essentially take the whole amount of DNA and how much DNA is in the comet, and that will let you know if a lot of DNA damage is occurring. So essentially, the higher the olive tail moment, the more DNA damage is occurring in the cell. And so these are results from HACAT cells at 7 weeks. And so I found that after 7 weeks of arsenic exposure, chronic arsenic exposure, that there's a significant amount of double-strand break accumulation occurring in HACAT cells compared to cells that are unexposed. In KIR T cells, it's a little bit different of a story. So when we look at overall double-strand break accumulation between unexposed and exposed cells, we don't see a difference. However, if you look at a subpopulation, we can see that at least in this higher damage subpopulation, more double-strand break accumulation is occurring. And this is at the 8-week time point. If I go all the way out to 32 weeks, then we do see a significant accumulation of double-strand breaks occurring in the chronically treated cells. And so this was good. This established that, we in our toxicologically relevant concentrations of arsenic, that we are getting double-strand break accumulation occurring. And so the question is how? And so I just want to do a quick overview of ATM activations. This is that apical kinase. And so double-strand breaks that can occur just at basal levels. This is that 10 to 50 double-stranded breaks that are occurring every day. They normally activate ATM. And so ATM activations marked by this phosphoserine 1981 modification. And so the ATM phosphorylation is cyclical, and I'll dive into more of that a little bit later on in the presentation. And in addition, we looked at ATR. And so this, although I didn't mention it earlier, this was another hit in terms of DNA damage dysregulation in the cells. ATR is typically associated with UV stress, and so this is why it's another interesting candidate for us to look at. And so ATR activation is actually marked by a different modification, phosphoserine 428, and it also has a cyclical phosphorylation and dephosphorylation occurring. And sorry, forgot to mention this. So downstream of ATM again is CHEK2, which activation of CHEK2 is marked by this phosphothreonine 68. And although it's not diagrammed here, it also goes through phosphorylation, dephosphorylation cycles. So these were our first exciting results. And so what we've found, or what we're looking at here really, so at the top is ATM activation marked by that phosphoserine 1981, and we're comparing it to total ATM. And so if we look over here where we've quantified our immunoblotting, we can see that there's significantly reduced activation of ATM occurring in cells that are exposed to arsenic. And when we look downstream of ATM at CHEK2 activation, marked by this phosphothreonine 68, compared to its total CHEK2, we can see that there is significantly reduced CHEK2 activation as well in the cells. So in essence, what this is showing us is that ATM signaling is down in cells that are chronically exposed to arsenic at this early time point in transformation. And so we also looked at ATR activation and total, compared it to total ATR, and we found that there were no differences. And so in general, this, these are our first results. So if we move on into Keratinocyte T cells, we see the exact same phenomena occurring, where if we look at ATM activation, there's a significantly reduced activation happening, as well as its downstream CHEK2, whereas ATR signaling is unaffected. And so these were really exciting results because it described or showed us a phenomenon that might be directly responsible for why double-strand break accumulation is occurring in cells. Okay, so what I just described is that we have reduced ATM activation after chronic arsenic exposure for 2 different human keratinocyte cell lines. And so since we know that arsenic induces accumulation of double-stranded breaks, one of the things we wanted to test was whether or not neocarzinostat, which is an agent known to induce double-stranded breaks, could also cause issues with DNA damage response activation in these same cell lines. And so for these experiments, what we did is so either HACAT or Keratinocyte T cells, we actually took one plate, divided up into triplicate plates, and either treated them with 20 nanomolars of NCS for 30 minutes, which essentially has been shown through different empirical experiments that we've tested to cause double-strand breaks. And so when we look over here at these immunoblots, I think it's pretty obvious to the eye that when we do treat cells, this at the top here is HACAT cells. When we treat cells with neocarzinostat, we can see a good induction of that ATM activation as well as downstream CHEK2. And the same thing occurs in Keratinocyte T cells. Here we haven't quantified the CHEK2 activation simply because it was an all or nothing. Pretty easy to see. And so what this is showing us is that these pathways are indeed active in these cells, and that arsenic is somehow acting to reduce activation of the ATM signaling pathway. And so the next question we have was, how does arsenic decrease ATM activation? And so I talked about ATM having sort of a cyclical phosphorylation, dephosphorylation. And so the things that are responsible for that are phosphatases. So these are proteins in the cell that dephosphorylate or remove this phosphate from that serine 1981 position. And so if we had an uptick in their activity, we could very well see what we're seeing. We could see that reduced ATM activation occurring. An alternative explanation could be that the initiator, or the MRN complex, might not be initiating or phosphorylating ATM as well in the presence of arsenic. And so that's the questions we sought to answer. And so we had some hints from our RNA sequencing data that expression for these 2 phosphatases, or some of their subunits, were actually increased in chronically arsenic treated cells, HACAT cells. And so that's the first one we went with. And so when we look at HACAT cells, we actually saw, opposite to what we predicted, or opposite to what our RNA sequencing showed us, we actually had reduced protein levels of PPP2R2C. This is a catalytic subunit for PP2A. And we saw no changes in PP5 protein levels. In Keratinocyte T cells, we saw a decrease in both of these with chronic arsenic exposure. And so one of the reasons this might be where we see a difference between our RNA sequencing data versus the data here, there could be post-transcriptional mechanisms that are regulating these mRNAs. So we also looked at miRNA expression, and we've identified 3 or 4 mRNAs that could directly target these phosphatase mRNAs. And so that likely might be an explanation for why we're seeing what we're seeing. So the next idea was to look at this initiator complex. And so when we look at the MRN complex, expression of the different components that make up the MRN complex in HayCat cells at the 7-week time point, we see RAD50 levels, although they don't significantly change, they are trending downward. MRE11 doesn't seem to change at all, and NBN1 actually increases. And so the only thing that we can really say about this is that the MRN stoichiometry is dysregulated in the presence of chronic arsenic exposure. And in Keratinocyte T cells, we looked at the same proteins. We actually found that in this model, RAD50 protein expression was significantly reduced, as well as MRE11, whereas NBN, although trending downward, wasn't significantly reduced. And so again, it looks as though chronic arsenic exposure disrupts this MRN stoichiometry. You do need a 1-to-1-to-1 ratio of these proteins in order for the complex to work correctly in the cells. And so in terms of my conclusions, I hope today that I convinced you that arsenic exposure causes double-strand break accumulation by showing the neutral COMET assays. I definitely think that our data are strong for showing ATM, reduced ATM activation occurring. In terms of the mechanism, I think the PP2A, PP5 story might be interesting. We do have some plans to look at phosphatase activity because, again, we just looked at protein expression, so we know nothing about their activity. And a lot of my F32 application is focusing on this MRN complex and how it might be affected by chronic arsenic exposure. And so, and I also showed downstream check too. So this, I think, was really a good thing to show that we actually not only see reduced ATM activation, but the thing that it's responsible for phosphorylating is also reduced significantly. And so this could be a reason for why we see cell cycle checkpoint disruption or problems in double-strand break repair occurring in cells that are chronically exposed to arsenic. And so one thing that I know I didn't really cover is DNA PKCS. I kind of left that player out. I do have plans to study DNA PKCS in the over the summer, actually, with a medical student. So, and with that, I'd just like to acknowledge all the lab members. Chris Dates, my mentor, has been awesome over the past year and 4 months now. And so I also want to thank the department in general. I came into the department and really at the University of Louisville at kind of a weird time, right in the middle of the pandemic. And so it's been just really fun getting to know everybody, even though it's been mostly virtual. But I'm glad that things are getting back to normal. It's been a really welcoming environment. And I'd also like to thank all my lab members, Mayuk and Anna, who have helped me considerably with manuscript preparation, experimental things in the lab. Sven, our technician and lab manager, who's helped with all the things in the lab that seem to just take up time. So thank you. And Lakin, my medical student last year who helped with a lot of the experiments. And of course, Bert Watson, who has attended our lab meetings and given really good critical feedback for our experiments. And then finally, my funding sources. And I'd be happy to take any questions. And